Perfect. Okay. Right, guys. So welcome to our paper 1 revision session. So the paper 1 exam is on Friday. The focus here is the higher tier content today. And as I said, I'm going to whiz through as much as I can. I will quite likely go over the hour, but I am going to try and talk as quick as I can. All right. So I'm actually working on PDF today because of a technical issue earlier on. So don't panic that the fact that it's a PDF and it's not a PowerPoint. Okay. Right, first things first then here. This will also help you with your physics exam as well. If I can make this a bit bigger, that will... How is that, ladies? Can you see that okay like that? Is that better?
Yeah.
Perfect. Okay, so when doing converting, so you know that in the first topic, which is the cell biology, we're going to have to do some calculations related to magnification. Okay, and so when you do your conversions, when you're converting from centimeters, so think about centimeters on a ruler, and then millimeters. For centimeters to millimeters, you've got obviously 10 of them. So to go from centimeters to millimeters, you're multiplying by 10. And then smaller than a millimeter, if we chop a millimeter up into 1,000 little pieces, that is down to a micrometer, and that is a 1,000 times multiplication to go from millimeters to micrometers. Okay, and then the opposite, obviously, going the other way for conversions. This is quite important for our magnification calculations in just a minute. Down the bottom, you have, when we're looking at more masses of things, we've got grams, micrograms, nanograms, and then picograms. Okay, and each time you've got from grams to micrograms. I tend to remember it one way, and then so that I don't have to remember both. Grams to micrograms, sorry, milligrams even. You multiply by 1,000, then you're multiplying by adding on 1, 2, 3, 4, 5, 6 zeros to go from what did I say, milligrams to nanograms, and nanograms to picograms again, times it by 1,000 again. So just know and be comfortable with converting units at higher tier. It will be one of the things that they're expecting you to be able to do, and will help you with your physics as well. Okay, do feel free to you can just shout out your questions or pop them in the chat box if you want to as we go. Okay. Right, standard form is the other maths thing as well here. Okay, and I think I'm quite confident that both of you are quite confident with your maths, but if you've got a number of what we got, 153, and you've got all these zeros here, with your standard form, you're putting your decimal place in, and then you're counting how many times have you moved your decimal place to figure out whether you've got what your power of 10 is here. Okay, and let's get this right. If you're moving towards the right, no, let's get this right. If you're going from here, this side of the 0, all the way to this side of the 0, it will be something to the power 5, as in a positive power. Whereas if you're getting a very small number, it will be a negative power there. Okay, I'm hoping that makes sense. My maths is not my forte. Okay, so point being, be comfortable with your standard form. I'll quickly do this bottom example for you to make sure I've explained it correctly. If you've got 11 and all of these zeros here, you go to 11, that's where your decimal place would be. Let's count how many places across. 1, 2, 3, 4, 5, 6, 7, 8 to get it to the end. And so that means you've got 11 times 10 to the power 8 in standard form. Hoping that makes sense. Okay, yeah. Brilliant. Okay. So now the topics that we're going to be trying to cover today. So we've got big plans for Friday, and hopefully again most of this revision is done, but we're looking at chunking this up, and this is slightly different to the revision guide in terms of how the topics are laid out, but these are a lot of the key topics, and some areas I will spend more time on than others today. Okay, so we've got in a minute, we'll look at the basics of animal cells and plant cells and whiz through that, but we've got cells, tissues and organs. And so we've got organization, we're looking at the cell cycle, digestive system, circulatory system, lungs and breathing. This encompasses sort of topics 1 and 2, a lot of, okay. Topics 3 is this all to do with disease. Okay, communicable and non-communicable diseases. We'll have a look at those with specific examples of diseases you need to know about. And then the last section is your bioenergetics, which is your topic 4, and that is your respiration, and that is your photosynthesis. And then we'll move on to a different set of slides for your required practicals, which we'll do separately at the end. Okay, are you guys both okay to hang on at the end afterwards if you need to, if we go over by sort of half an hour or so, possibly?
Yeah.
Fantastic. If you have to go, just pop it in the chat box and let me know. That's absolutely fine. That's not an issue. Okay. So again, basics, your animal cell and your plant cell, these are both eukaryotic cells. Okay, and all living organisms are made of cells. You have your common features of your animal cell, or the features that are common, sorry, to animal cells and plant cells. So you've got your nucleus that's in both, your cytoplasm at the top here that's in both. Okay, your ribosomes tend to be in both, mitochondria tend to be in both, and your cell membranes are in both as well. The plant cell has additional features. It has additionally a chloroplast or chloroplasts, where photosynthesis happens, a vacuole in the middle where the cell sap is stored, and then the cell wall around the outside, which gives it support and helps it keep its structure. Okay. Other things, the nucleus that we mentioned earlier on, this is where the genetic information is kept and it controls the activities of the cell. That's the language we want to be using. Okay. The cell membrane controls what can go in and out of the cell. It's almost like it's like the bouncer at a party. I always teach it like that. So it decides what goes in and what goes out of the cell. The mitochondria releases energy through respiration. Please take note, not produces energy, releases energy. Okay. Do not talk about it as the powerhouse of the cell. I don't know where that came from many years ago, but it's incorrect. Okay. Mitochondria, you could just say it is the site of aerobic respiration, right? Or it releases energy via aerobic respiration. That works. So with the cytoplasm, so this takes up most of the space of the cell, both in the animal and in the plant cell. This is where most chemical reactions take place in the cell. And the ribosomes are these little dots here that you see around the cells. This is where protein synthesis happens. Additionally, I've already mentioned the cell wall. It strengthens and supports the plant cell. Permanent vacuole is this massive thing in the middle of the plant cell. This is filled with cell sap. It helps a plant cell to keep its cell structure. Okay. Chloroplasts are these green organelles that are floating around the outside. This is the site of photosynthesis. They contain this pigment called chlorophyll. All right. Remember, these are both eukaryotic cells because they have a nucleus and their DNA is within that nucleus. Okay. They're complex cells. Okay. Now, so a couple of definitions when we're moving on to sort of organization. Okay. The microscopy stuff we'll cover in a minute when we get to the required practical stuff at the end. Okay. So don't think I've forgotten that. So a tissue is just a group of cells that work together to perform a similar function. That definition you're going to need to know sort of word for word. I'm sure you already know it. Okay. An organ is the next level up. So what we talk about is if you're thinking about going smallest to biggest, you'd go cells down the bottom, then you've got your tissues, then you've got your organs, then we have our organ systems, then we have our organism at the top. Okay. So our cells come together and they form tissues. So the group of cells doing a similar function. Then your tissues come together to form an organ. So an organ from the board is a group of tissues that work together to perform a similar function. The structure of the answer is the same. Okay. Our examples of tissues, just think about where would you find a whole bunch of cells doing the same thing? Blood. So blood cells, right? So blood is actually a tissue because you've got lots of blood cells in there. Muscles made of muscle tissue, epithelial tissue in the small intestine is another good example, and made of epithelial cells. Organs, examples of organs, this should be a bit more obvious. The heart, the liver, the stomach, the pancreas, the lungs, etc. These are a group of tissues that are doing a similar function. Okay, so that's two important definitions here. And then this will keep coming back to this slide so we can stay on track. Okay, cell cycle. Okay, so this is with the cell cycle, this is what we're talking about when we're talking about mitosis and meiosis. And in a minute, what we'll do is we'll do a nice comparison of both of these. What I am going to do super quickly is open up the slides on this computer as well, so I know to track how far through we are. Perfect. Okay, so the cell cycle. This is where cells divide for growth and repair. Okay, so if the question in the exam says, what's the function of mitosis, for example, we're talking about growth and repair. That's the only reason you'd have mitotic cell division. With this heading should say mitosis, I think up the top here actually be more appropriate. So with mitotic division, the genetic information from the parent cell is exactly identical to the genetic information in the daughter cells that are formed. Okay, the main stages are the cell begins to grow. Okay, so the cell is preparing for division. Secondly, DNA synthesis. So this means that all the chromosomes are now doubled. So they change from looking like a sausage to looking like double strands here. Okay, this is DNA synthesis. Again, this is the preparation for the cell, one cell to divide into two. Okay, the DNA is then checked for errors. Okay, fine, because we've got to make sure that we're copying our DNA correctly. And here at stages 2 and 3, you also have the replication of all the organelles that are in the cell. So things like mitochondria, things like ribosomes and other organelles in the cell are also copied, because remember, we want our parent cells to be identical to the daughter cells. Then you have the actual mitosis bit. So this is the diagram that you would have put into your books or on your flashcards, where you've got one cell, you've got that's one cell again underneath, and then you've got it splits into two at the end. Okay, and so that's the actual mitosis bit. Okay, but everything else at the beginning, 1, 2, and 3 is preparation for mitosis. And then at the very end, after mitosis has happened, the cell actually stops dividing. Now, the cell has to stop dividing, and if it doesn't stop dividing and there's an error in the dividing signal, this is what causes cancer, which again, we're going to touch upon later on in the session. Okay, so here we go. And so here we have, again, the word for word key, what we've got. Okay, so mitosis in a nutshell. The DNA replicates to form 2 copies of each chromosome. Already said this. The nuclear membrane around the DNA starts to break down. The chromosomes then line up down the middle of the cell. So you see all these X's down the middle of the cell. The chromosomes then are pulled apart to each end of the cell. And that's the image where you see, to me, it looks like seagulls that are like flying to each end of the cell, okay, like little V-shaped seagulls. The cytoplasm and the cell membrane then divide at the very end, and this is what produces your diploid cells at the end, okay? When we're talking about diploid in humans, we are saying we're talking about a full set of chromosomes. That means in humans, that is 46 chromosomes. Okay, and because at the beginning of mitosis, we doubled our 46, we formed it for momentarily, we formed 92 chromosomes because we're doubling 46, and then the whole cell then split. Okay, once the chromosomes were divided and cytoplasm and cell membrane are divided, and because you've already doubled it, at the very end, each of the new cells that you make also contain 46 chromosomes, exactly like the parent that you started off with. Okay, we're producing identical cells, really important. Remembering mitosis is for growth and repair. And so this is for instances like when you break a bone, when you cut yourself, when you, I don't know, when you scrape your skin on something and we need to make new cells, that is when mitosis happens. Okay. Okay. Moving on now to meiosis. Now, oh, actually, I should come, just let me just up here with mitosis, just to remind you, with mitosis, you start with one cell and you end up with 2 cells. Okay, so when you see a diagram, that's exactly what you're looking for. One cell going to 2, that's mitotic division. Okay, now with meiosis now, okay, this is where you are making either sperm cells or you're making egg cells. Okay, the function of meiosis is to produce gametes or sex cells, sometimes they're called. In plants, we would talk about egg cells and pollen cells. So the female gamete in plants is egg, and the male gamete in plants is pollen. Okay. So again, the steps are broadly the same. Okay, you have chromosomes that make identical copies of themselves. Chromosomes are copying just like they did in mitosis. Similar chromosomes pair up. So chromosomes pair up in what we call homologous pairs, and that means they each, like chromosome number 1 has a matching chromosome number 1, chromosome number 2 has a matching chromosome number 2, and they try and line up next to each other. We also have a new thing that happens in meiosis is that sections of DNA can get swapped. So where you have 2 chromosomes, I might actually write on the slide to show you, where you have 2 chromosomes next to each other, what you can have, if my board's going to let me write, here we go, you can have your first chromosome here and the one next to it, will be next to it here. And these two bits, look, I'm going to just kind of draw them longer to show you. This is the crossing over bit that can happen. Okay, they obviously won't get longer, they'll be set like that. But in this region here, you've got like the two dangly legs, if you like, of the chromosomes that are crossing over. And what can happen is those sections of DNA can eventually get swapped. So if we call this chromosome A and call this chromosome B, rather than just being all A and just being all B, after the crossing over bit, A will have a little bit of B on the end and B will have a little bit of A on the end. Okay, and this swapping or crossing over is one of the things that causes variety in meiosis. Okay, so meiosis produces variation. And so that's this idea. And again, if you're not sure, pop into your revision guide and it will show you what this crossing over looks like. Okay, and then nextly, the last point here, the cells actually divide. This is really important. They divide twice to produce haploid cells. And so again, I'm going to write it down here because otherwise I'll forget to tell you. Your diploid number in humans, you've got 46 chromosomes. And with the haploid number, think of the HA like half. Okay, and so the haploid is, whoops, if you can spell it, not like me, haploid, there we go. The haploid number in humans is 23 chromosomes, so half of this. Okay, chromosomes. And the only cells ever in humans that are haploid are the gametes or the sex cells. And this is going to be in males, you're going to be talking about sperm. And in females, you're going to be talking about eggs. Okay, again, in plants, you're talking about eggs and pollen. All right, everybody okay, girls? Everybody all right?
Yep.
Yeah.
Fab, thank you. Just checking in. Can I get rid of the annotations?
Yes.
Perfect.
Yeah.
Okay, perfect. Right, okay, so, and then I'm going to actually get rid of you, Minnie, as well. Okay, perfect. Right, how's my pace, ladies? Is it working for you?
Yeah.
Yeah, it's good.
Good. Okay. So I'm hoping a lot of it you're thinking, oh yeah, I knew that. Oh, but maybe there's this bit that I've forgotten. That's the whole idea. Okay. Okay. Transport in cells. So now we're talking about how does stuff move within and across cells. Okay. We're just talking about how stuff moves. So there's 3 ways that you need to know. So you've got, first of all, the most straightforward one is diffusion over here. And diffusion is where you've got molecules that are moving from a high to a low concentration. So in your head, you're picturing me stood next to you, or maybe actually we're in the same room, you and you're at one end of the room and I'm at this end of the room, and I spray myself with this really stinky perfume, okay? And then what's going to happen is I've then got a high concentration of perfume molecules around me. And eventually what's going to happen is, so those perfume molecules are going to want to start to move around and they're going to want to spread out, because that's what diffusion is, this passive spreading out of molecules. Eventually, if we both stay still long enough, those particles are going to eventually spread out across the room and you're eventually going to be able to smell that perfume and then tell me how horrible it is. Okay, so it's a high concentration to a low concentration, and we call this a concentration gradient. So I want you to imagine across this room, if I'm stood here and you're stood across the room, if I've got lots of perfume particles on me and we draw like an imaginary graph across the room, like a line graph, we would draw it as a really, really high line where I am, because there's lots of perfume particles, and then a really, really low line in the beginning where you are, and then you've got this gradient, what we call a concentration gradient, that helps those particles, that shows, sorry, I've got lots of particles near me and you have very few particles near you, a low concentration. And what that also means is that the bigger that difference, i.e. the higher the concentration gradient, the more easily or the greater rate there is going to be of diffusion. Okay, so if I spray half a bottle of that perfume, okay, we've got a very, very high concentration at this end, and so of perfume particles, and so you're going to be able to smell those particles sooner compared to a low concentration on me. If I do like 2 squirts, you might be waiting ages to smell it. So key things about diffusion: it does not need energy, okay? It is a passive process, and it also does not generally involve a membrane, okay? And so what happens is this diagram down the bottom explains it nicely. You've got a high concentration on one side here, a low concentration on the other side here. And as time passes by, eventually you get to a point of equilibrium, which means that those particles have spread out pretty much evenly. Now they're still moving, okay, but you've got, say you've got like 3 moving from one side to the other, you'll have 3 moving from that side back again. Okay, so we call this a dynamic equilibrium in biology. Diffusion, just to take you back to key stage 3, happens in liquids and in gases. Okay, because you've got movement of particles in both those states of matter. Next way of transporting stuff is osmosis, which is the middle box. Osmosis is how water molecules move, okay? And osmosis is always specific to water, okay? In this case, in your definition, you're going to talk about a semi-permeable membrane, okay? Which is a membrane that lets, it's just in the diagram down here, it lets some stuff through, not all stuff, okay? Again, you've got movement from a high water concentration to a low water concentration. Okay, so this requires you to think a little bit back to front, okay, because the words are still the same as diffusion. However, where you've got a lot of water, you will have very little what we call solute. Okay, so if you have a look in the diagram down here, you've got this red line is your semi-permeable membrane, and you can see on this side here with this one sugar molecule, you've got a lot more water. Okay, and on the other side, you've got, compared to this side, you've got a lot more, sorry, you've got a lot less water. Okay, and so let me just, yeah, so the blue little arrows actually show you've got water moving both sides, but look what's happening. Over time, you've got water that has moved from this side. It's not a great diagram to describe it, but you've got more water that's moved from the, as you look at it, let's have a look, your left to your right, because there was more water available compared to solute in the initial diagram. I hope that makes sense. Okay, again, osmosis is does not need energy. It is also a passive process. Okay. The common questions, again, there's a required practical on this to do with the potatoes. This came up several years ago. So you would have learnt about osmosis and water moving in and out of potatoes. You did the practical in school with the little chunks of potatoes that you cut out with like a cork borer, we call it, or an apple corer. A little tube of potato, you put it into different salt solutions, okay, different concentration of salt or sugar solutions it could have been. And then you looked at the mass before you put it in, and you looked at the mass of the potato after the experiment, after leaving it in the solution. And if the mass went up, we would explain that by saying the mass must have gone up because water has entered the potato by osmosis. Okay, that also is a good experiment where when you graph your results, you will get a line graph. Okay, and on that graph, if they ask you, quite unlikely to ask you, but the line graph of results where you've got concentration going across the side axis, and then the mass up the side. Sorry, concentration along the bottom and mass up the side. You plot all your points of your change of mass, okay? And then there is a point at which at 0, okay, where the axis comes across, your line will cross the 0 axis. That is the point where you could say that the concentration of sugars in the potato is blah, 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 whatever point of concentration, your graph line crosses your concentration line. If you're not sure, at the end, I'll come back to it and quickly sketch it out for you. It's in my head, but clearly, because I've not put it on the slide, you might not be able to visualize it. Okay, active transport. This is the third way that stuff moves in and out of cells. So this is the opposite of diffusion. So this is where it's like you walking down a corridor and there's 20 people coming in the opposite direction and there's just little old you trying to get through them. Okay, you are going against the concentration gradient. You're going from where there's a low concentration of people, you, to where there's a high concentration of people, all those other people. Okay, so it's low to high concentrations active transport. Because it's going to be really tricky for you to push past all those people, you're going to need to have energy to do this process. Okay. It more often than not, no, not more often, pretty much always actually, would happen across a membrane. Okay. And in this diagram down the bottom, you've got a membrane here, which is what these little ball things are here. You've got inside and outside the cell, could be the other way around. You've got very few of these green molecules on one side. You've got loads on the other side. So if we were just talking about diffusion, we'd be moving from all of those green particles, the high concentration, to the low, but we're not talking about diffusion. We're talking about active transport. And this is where you have one or two molecules, low concentration, and those particles are moving across this particular channel into this side where there's lots of them. And that could be for lots of reasons. In this case, probably because these green squares are needed for another reaction somewhere else in the cell. Okay. Right. So that's how stuff moves. Let's go through. Okay. We'll keep moving through. Okay. So your digestive system. Now, you need to be able to label this journey of food down your digestive system or through your digestive system. So labeling each part, and they might ask you to just fill in a blank somewhere, quite a standard demand question as opposed to higher. We're going to go through now and so you'll be able to recognize them. But you want to, in the labeling, you're talking about probably a sort of a grade 4-ish kind of level. If you can tell me what each of the parts does, so the functions of each of the structures, then that then starts pushing you up to a 6, 7 and above. Okay, so when food enters your mouth here, you've got the food, you've got physical digestion in your mouth that's breaking down your food into smaller pieces. You've also got some chemical digestion because the salivary glands here are starting to release digestive enzymes like amylase and starting to break down some of the food in your mouth. The food then travels down your esophagus. So this is the path that the food takes to reach the stomach. So the esophagus goes down here. Your stomach is a sac here that does, again, physical churning of food because you've got muscles in your stomach. Okay, you've also, as you know, sorry, I've got a very itchy nose. You've also got acid in your stomach, very, very acidic conditions. And that acid can have a pH of 1 or 2, super, super acidic. And again, physical and chemical digestion of food happening here. Then after your stomach, let's go, we'll come, let's talk about the liver now. The liver is here that stores, stores your bile. So imagine it like a little box of Tupperware storing the bile. Sorry, let's get the right way around. The liver produces the bile, okay, so the bile factory. And then the gallbladder is actually where your bile is stored. So your Tupperware is this tiny little gallbladder here. Okay, remember we need bile to help break down fats in our diet for emulsification of fats in our diet. Okay. The pancreas. So after the stomach here, we're now reaching this leaf-like structure called the pancreas. And the pancreas is responsible for producing hormones that help you control your blood sugar. Okay, and this comes up again in paper 2. And that recognizable by the leaf shape. Okay. After the pancreas, again, we're following down, we get to the small intestine. This is where the nutrients and all the good stuff from your food is absorbed. Quite a long, thin tube. Imagine it like a whole heap of sausages all squashed together in your abdomen. Okay, your lower abdomen. After the food passes your small intestine, it comes round here and passes around the large intestine, which is like a much, much bigger tube compared to the small. And the large intestine's function is to reabsorb water from your food. Okay, and so as this bolus of food passes through the intestine and large intestine, again, we're talking about peristalsis. And peristalsis is how the food actually from the stomach will move. It's this muscular movement of this. If you imagine the food that's in your stomach that's passing through your digestive system like a ball of food, okay, we call it a bolus. And it's like putting a tennis ball into a pair of tights, right? And then I'm going to ask you to push it through. That's what peristalsis is. It's a muscular movement of those tights that will push that bolus of food all the way through your digestive system. And we said when we get to the large intestine at the very end, okay, this is where you form your fecal matter, your poo, right? And in your rectum, your poo is actually stored until it's egested via the anus, okay? And that's the kind of whistle-stop tour of your digestive system. Okay, so Some other, let's go a little bit deeper into the functions of some key parts here before we move on to looking at enzymes. Is it enzyme? Yes, it is. Perfect. So in your stomach, I've already mentioned all this, so this is repeated again. You've got acidic conditions in your stomach. It contains an acid called hydrochloric acid. Okay. The function of the stomach is to break your food down. Now, it contains muscular tissue. And again, whenever we're talking about muscles, just to let you have it in your mind, any muscle in your body will appear, it will appear in a pair. Okay. And what happens is with these, what we call antagonistic pairs of muscles, you will always have one that's contracting and one that's relaxing. Okay. And then you might have the other contracting, the other relaxing. So biceps and triceps, in order to move, you will have a contraction and a relaxation, contraction, relaxation, right? So this antagonistic pairing is how muscles work. And in the stomach, you've got muscular tissue that contracts and relaxes to help you churn your food, this physical, or we can actually call it mechanical digestion of your food. Within the stomach, you also have epithelial tissue, and this is the tissue that lines the stomach. And the question I always get asked when I teach about the stomach is, why doesn't the acid come out and digest the rest of the body? Well, the reason is the acid in the stomach is contained within this epithelial tissue that is really good at lining the stomach to protect it against being kind of broken down by the acid. Lastly, you've got glandular tissue. And when we're talking about glands, we're talking about the organs that produce hormones. Okay, that's their functions. Again, paper 2, looking more at hormones, right? So what does glandular tissue do? It secretes digestive juices and enzymes. Okay. So bile. So bile, again, I touched upon this earlier on, is an alkaline substance, okay, which it's helpful because as food comes out of the stomach, excuse me, into the gallbladder, the liver and the gallbladder, as the bile is produced, okay, it becomes neutralized. Sorry, it neutralizes the acid from the stomach. So this bolus of food passing through, any residual acid from the stomach is neutralized by the bile. And the other job of the bile is to emulsify the fat. Now, emulsification is where you've got these big chunks of fat, if you like, and we are breaking it down into tiny, tiny droplets. And those tiny droplets have a much larger surface area than these big gloopy bits of fat. And so it allows those fats to be broken down a lot more easily. Okay, so emulsification is just producing, it's the same, the test we'll look at in a minute, the food test for fats is when you put ethanol in and put water in and shake it, it goes from clear to like cloudy because emulsification has taken place. These tiny drops of fat are spread out all through the liquid. Now, the small intestine down here. We've got, again, I said to you, it's like a massive long tube of sausages, okay, a long string of sausages, all squashed into your lower abdomen. So the small intestine's got a large surface area so that it can absorb the molecules, which means your food that's passing through your small intestine, all the good stuff, all the nutrients, the vitamins, minerals and stuff, is being reabsorbed into the bloodstream through the epithelial cells of the small intestine. Once that food passes out the small intestine, look, it's surrounded by blood vessels. So it's blood vessels all the way around this small intestine. And this means that all that good stuff, the soluble molecules, can be transported to other parts of the body. Okay, really, really, really important function. And this is why nutrition is so important as well. Okay, let's go on to enzymes. Now, I would put money on it you're going to get an enzymes question on Friday. Okay, in some way, shape or form, they will talk about enzymes. They're so important and it's such a big chunk of this particular topic. So remembering the language of enzymes, first of all, okay? And by the way, if you get a 6 mark on this, you guys are just absolutely laughing. I'll just keep my fingers crossed that you get a 6 mark on this and just know this structure really well. So enzymes have an optimum temperature and optimum pH. So that is the temperature and pH that they like to work at the best, okay? If the conditions are not correct, so if we are heating up our enzyme too much, if we are changing the pH away from the optimum temperature or pH, the enzyme becomes denatured. And now this has vaguely said this, and I'm going to be a bit more specific with how you're going to answer a question on this. If the enzymes denature, it means that their active site changes shape. If the active site changes shape, the enzyme is no longer, the shape of the active site is no longer complementary to the shape of the substrate. So we cannot form an enzyme substrate complex. If we can't form an enzyme substrate complex, we're not going to produce a product. In other words, the enzyme stops working. Okay, so that's the key to an enzymes question. Again, enzymes denature, active site changes shape. Okay, active site is no longer complementary to the substrate. Shape's different. We cannot form an enzyme substrate complex, and therefore product is not formed. So when we're talking about digestion, we link this in beautifully because there's 3 key enzymes that you need to know about. You need to know their names, you need to know where they're formed and where they do their job. Okay, so at the top here, we've got amylase. And this is usually, I think of this like in your saliva, because salivary amylase is the big one. Another word for amylase, excuse me, is carbohydrate. Okay, because not amylase, glucose, which is a substrate that it works on. Sorry, just to get this right, starch, which is a substrate that this works on. Okay, imagine starch like a really long chain of paper clips. It's a really, really big molecule. In our food, we get starch from things like bread, from cereal, from pasta, like all that starchy, like very carby food. Potatoes, another big source of starch as well. So what happens? The amylase enzyme works like Pac-Man and breaks down the starch, this really, really long chain of paper clips, and it chops that starch up into single paper clips, which we would call glucose, right? And you know glucose from photosynthesis, C6H12O6. So, second enzyme is lipase. Okay, lipase works on lipids. The lipids is a fancy word for fats. And lipids, when they're chopped up and broken down by lipase, they are producing fatty acids and glycerol. Okay, and lipase is produced, actually, I should mention the previous one. Amylase is produced in the salivary gland, the pancreas, and small intestine, 3 places. Lipase is produced just in the small intestine. Okay, and they've put here again, higher tier focus, a lipid is made of 3 fatty acids and 1 glycerol molecule. Last enzyme is protease. This breaks down proteins, as the name suggests. It's made in the stomach, the pancreas, and small intestine. Think about your proteins. I think about these like a big chunk of Lego that you've all stuck together. If we break off those bits of Lego, each Lego block would represent an amino acid. Okay, so proteins are made of amino acids. So think of this starch, lipids, and proteins as the big thing, and then the glucose, fatty acids, and glycerol and amino acids as the little chunks that it's made of. Okay, so where are we at? We're just here. Oh, we've got quite a bit. Okay, let's talk about the lungs then. So again, structure and function. Humans need organ systems for exchange of materials. They've got a small surface area to volume ratio. So you need to know the structure of the lungs and be able to label. Now this diagram's just come from bite size, key stage 4 bite size, but go through and know the labels around the lungs. And then hopefully the next slide, yep, we'll go into a bit more detail about these alveoli at the end. This is what we'll spend our time on now. Okay, so the alveoli is a place where actually the exchange of gases happens. So it's where after we've breathed in oxygen, it's where the oxygen goes from the lungs into your blood, and then breathing out is where our carbon dioxide leaves. And what's happening is the blood has carbon dioxide in it that's been moved into the alveoli, and then the carbon dioxide is breathed out into the air. Okay, so again, review this. Again, you'll have the recording. By all means, pause it and come back and just remind yourself what it's all about. Now, the alveoli. This is the structure at the very end, and I'll just show you in the last slide. I just pointed at it. here, so if you follow your bronchioles and your bronchus and bronchioles all the way to the very end, these little kind of little ball things at the very, very end of your lungs, these are your alveoli. And they are covered, you can see here, with blood capillaries, okay? They look blue and red, and we'll talk about why that is now. So where the gas exchange actually happens, we're going to take one of these alveoli and slice it open. In the alveoli, you can see you've got all this space in the middle. Your air's coming in that's hopefully full of oxygen, has got lots of oxygen in it. And the air that's leaving, again, hopefully has a lot of carbon dioxide in it. Your alveoli is the actual ball here. And around the outside, you've got a blood capillary where you've got blood flowing constantly past the alveoli. And as those red blood cells come along in this blood capillary, they will drop off the carbon dioxide that they have got from the cells that have been doing respiration. We call that deoxygenated blood because it's carrying carbon dioxide rather than oxygen. And then as they pass across and around, I should say, the alveoli here, the red blood cells, as they drop off their shopping bags of carbon dioxide, they're picking up their shopping bags of oxygen. Okay, and as they pick up their oxygen shopping bags here, okay, they become oxygenated, and then they can go off on their journey down the capillary, down the arteries. Because they're oxygenated, they're going to go to the cells that are doing respiration, okay, because they're the cells that need oxygen. Now, you might be asked, what are the what are the structures of the alveoli that help it perform its function? You could talk about the fact that the wall of the alveoli is only 1 cell thick. That shortens the diffusion distance. Okay, so we're increasing the rate of diffusion. They are lined with a moist surface, and again, the moist surface lining helps more diffusion happen. They're small to give, again, a large surface area to volume ratio. The bigger the surface area, the more diffusion can happen, or the greater the rate of diffusion, I should say it like that. There's a large number of alveoli in the lungs. Again, we're increasing the surface area to increase the rate of diffusion. And we're surrounded by a capillary network, which is easy for gases to diffuse across. So we're remembering here that as the blood cells are coming along and moving across, we're constantly maintaining a diffusion gradient. That means you will have, if you think about this again, higher tier, inside the alveoli, you will have a high concentration of oxygen, and so the oxygen diffuses from inside the alveoli to outside to the red blood cells. Inside, hopefully, the alveoli, you'll constantly have a low carbon dioxide concentration because you're hopefully constantly breathing it out. If you've got a low carbon dioxide concentration in the alveoli, it means that where the red blood cells here have a higher concentration, the carbon dioxide is moving from the red blood cells into the alveoli constantly. Okay, so the key process, the movement of the oxygen and carbon dioxide is by diffusion here, from where there's a high concentration to where there's a low concentration. Okay, right. Let's move on to the heart. Okay, I've got a feeling time-wise we might go way over, girls. Is this okay so far? Is it working for you?
Yeah, it's good.
Yeah, good. Okay, I'll just carry on through. As I said, if and when you have to go, that's fine. If we're a couple of hours, is that going to be all right? Because I think I've grossly underestimated how much time we need for this.
Yeah, that's fine.
Yeah, just let me know if you need to go. It's not a problem. I'll just carry on and pop you over the recording for anything that you miss. Okay, structure of the heart then here. Okay, now remembering, as soon as you look at the heart, first thing you're going to do, looking at the diagram, is you're going to go, right, where is the thickest ventricular side? The bottom bit here. This side here is the thick ventricle wall. That is your left side always. Okay, just remember it like that. So left side is a thicker wall, and then remembering, following on from that thought, if it's got a thick muscular wall, what that means is that we're going to have much, much higher pressure blood on the left side of the heart. So as the blood, I'll talk about the process going through in just a second, but thick ventricular wall, high pressure blood, if it's got high pressure blood, as it leaves the aorta here, it's going to have a really, really big push. It needs to get to the very tips of your fingers and very tips of your toes. And in doing that, we know it needs to go a long distance, high pressure blood. Well, then therefore it must be carrying oxygenated blood to the respiring cells. Now, path of the blood. On the left side, we'll stick to first. You've got the pulmonary vein. This is the only vein in the body that carries oxygenated blood. All the other veins carry deoxygenated blood. So the blood comes into the top atrium at the top of the left-hand side of the heart. As the atrium fills, the blood passes down through this valve here into the ventricle. And the atria actually contract, both the left and right, squeeze at the top to squeeze the blood downwards. As the blood gets squeezed downwards, you then have the second contraction, which is the ventricles at the bottom squeeze together and push the blood up and out. And so the blood comes all the way up here through another set of valves out through the aorta, remembering high pressure blood, oxygenated blood, and the two valves we've already said. Okay. On the right-hand side, everything is the opposite. So blood entering the right-hand side again. So first of all, let's go. Your thin wall on the right-hand side. Your vena cava here is where your deoxygenated blood enters the top atria. Then we've got the atrial contraction, so the top bit squeezes. Oh, this is important. In the wall of the right atrium, and it's labeled here in the middle, but it's actually more on the outside here, you've got a whole bunch of cells that we call the pacemaker. Okay, sometimes it's called the sinoatrial node, but you need to know it as the pacemaker. And this is the thing, the bundle of cells that actually initiate, or they start the electrical impulse that kind of goes over the heart to make it contract. Okay, so just imagine them like, I don't know what movie it is I'm thinking of, a kind of bzz, 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 bzz. It's constantly zapping and sending the electrical impulses to get the heart to correctly beat all the time. And on the right-hand side, again, we said the vena cava, blood entering the atria at the top. The blood passes down when the atria contract, down to the right ventricle, and then the blood passes up, okay, through the pulmonary artery, up and out, okay. And again, you've got another set of valves, and this is low pressure blood here, okay, on the right-hand side. So, okay. So key thing is know the structure, atria at the top, ventricles at the bottom. Know how pulmonary vein, blood's coming in, vena cava, blood's coming in the right-hand side, and then out, aorta, left-hand side, pulmonary artery, right-hand side. Again, the pulmonary artery is the only artery in the body that carries deoxygenated blood. All the other arteries carry oxygenated blood, okay? The valves obviously are really important because they ensure that the one-way flow of blood happens and there is no backflow of blood. I was going to say something. Okay. Now, if you've got oxygenated blood that is leaving the left-hand side, that is going to the respiring cells, as we've said, the deoxygenated blood on the right-hand side that is leaving the right-hand side, because it's blue and deoxygenated, well, the first thing it needs to do is to go to the lungs to make it drop off the carbon dioxide, as we said, at the alveoli, and to pick up the oxygen, and then it will come back round on the left-hand side of the heart. And so for this reason, we call it a double circulatory system. So what that means is if you track and follow one red blood cell from one side of the heart to the other, you'll find it passes through the heart 2 times. Okay, so it says one circulation to the lungs. So say this, we're tracking a guy from here, he will go to the lungs. And then it says one circulation to the rest of the body. So from here to the lungs, and then from the lungs, they'll come back in here once they're oxygenated onto this side, up and out the aorta to the rest of the body. So they pass through, the red blood cells pass through the heart twice. It says they're simple cells, or contain simple cells. They have a smaller surface area, so diffusion is enough. I don't know what that point's alluding to. The valves. Valves can become faulty and need replacing. So if they don't work properly, oxygenated and deoxygenated blood can mix, and then blood can flow in both directions. It's really, really important that both of our sets of valves are working correctly, and you can have surgery to improve the valve condition. Some vitamins, it's putting me up. Right, we said left-hand side. Yeah, the higher pressure on the left-hand side is really, really important. Okay, the oxygenated blood. Okay. Right, I've mentioned a lot of this already. Again, you've got arteries. Now these have a narrow lumen. They've got thick muscular walls. In my head, I'm thinking of, I need loo roll in the bathroom. So I've just picked out a new loo roll out the packet. You've got a small lumen, a nice big thick wall of tissue paper on the outside. They carry blood away from the heart via the aorta to the rest of the body. High pressure blood because it's oxygenated. Okay, cool. Your veins are the opposite. You've got much thinner walls. The lumen is much bigger. Okay, and you've got the blood isn't at such a high pressure in the veins. It's much lower pressure. And so because the blood is at lower pressure, we have the possibility that it could go backwards. And so veins, have valves to stop the backflow. Okay. Now, in between arteries and veins, thinking about the fact that both arteries and veins, nothing can actually pass through them, as in through the walls of them. You can only have blood actually flowing through the lumen. Capillaries are the connecting vessels between arteries and veins. And where you have capillaries, this is where you can actually, stuff can leave and stuff can enter the bloodstream. And so the structure of a capillary is tiny, tiny, tiny compared to veins and arteries. Capillaries are only 1 cell thick to shorten the diffusion distance. Okay, and so they're the 3 blood vessels we need to know about. Now, they might ask you, they might mention different types of cells in the blood. What is a red blood? What is red blood? What is your blood made of? So in your blood, you have red blood cells, which the function of which is to carry oxygen to the cells. You have white blood cells, again, part of your immune system, and they are there to fight any infections. You have plasma, which is that like gooey yellow stuff. When you spin out a tube of blood in a machine, you'll find the red blood cells down the bottom and this yellow gooey liquid that is plasma, and that makes up quite a lot of the blood. And there's all different stuff dissolved in that plasma, like amino acids and things like that. And then you have platelets in the blood, and this is what, when you cut yourself and you start forming a scab, that is what platelets are used for actually clotting the blood. Okay, just keeping track of my slides here for you. Lovely job. Okay, down the bottom, okay, some features of red blood cells. They have no nucleus, okay, and very few organelles, and this means that they can bind more oxygen as they go. Okay, more space for oxygen to be bound. They have a large surface area. Again, you've got that kind of, we call it a biconcave disc, like a Krispy Kreme donut without the middle pushed all the way through. Okay, that gives it a large surface area. That means more oxygen can bind. Okay, they contain this molecule called hemoglobin. And again, at A level, we look at hemoglobin more closely, but you just need to know it contains hemoglobin. That's all. Okay. Right. We're now gone to, okay, right, cool. Right, non-communicable disease. Are we okay, girls?
Yep.
Yep.
Yeah.
Yep, fab. Okay. So we're getting there. Now, I want to say, how far are we? Let's just get this planned out. What I don't want to do is take up your whole afternoon, but I will, as I said, I will keep going. Right. Where are we at? Gosh, I want to say we're only about halfway there. Okay. Right, I'm going to keep going. Is that okay?
Yeah, that's fine.
Cool. Until I run out of steam. I'll let you know when I run out of steam. So, a non-communicable disease. Okay, so this is a disease that's not caused by a pathogen, and so it can't be spread from one individual to another. If you look at the term there, non-communicable, okay, you can't pass it on. And so we're looking at examples of diseases. There's only a couple here, that's good. We've got cancer and CHD, coronary heart disease. Okay, so for each of these diseases, it is something that can develop according to certain risk factors. So if your lifestyle involves smoking, if you've got a lot of alcohol in your lifestyle, if you have cancer that runs in your family, okay, so there could be genetic risk factors. If you decide that you're going to go on sunbeds every week, or you're out in the sun sunbathing, you don't wear suntan lotion and you tend to burn your skin, these are all what we call risk factors for cancer. They're things that increase the chances of you getting it. Now, if you do get cancer, and nowadays cancer is common in more common, it's something like 1 out of 2 people will get some kind of cancer in their lifetime, which is a crazy statistic. It blows my mind. But you can treat it with chemotherapy. So that's delivering specific drugs to the tumorous part of the cancer. Also, you could remove the affected organ if it's not a metastatic tumor. Okay, and so if it's a tumor that is not actively dividing, and we know, for example, it's, I don't know, in the, oh gosh, I don't know, example, in the cervix, for example, say we've got a tumor in the cervix, we can operate and actually physically remove that the cervix, or the womb it would probably be, to try and now prevent any spread of those particular cancerous cells. A reminder that cancer is just a disease where the stop signal for division, mitotic division, is not working. So rather than stopping dividing, those cells keep dividing and dividing and dividing. That's what forms your tumor. Okay, and eventually that primary tumor, the first one, if we don't pick up the cancer early enough, the primary tumor will form, some of the cells can break off into the bloodstream and go elsewhere and form a secondary tumor. And that secondary tumor becomes a lot more difficult to treat, because it might be that it's not just one in another place. It could be there could be a couple of others as well. So we need to really track, and this is why early detection for cancer is really, really important. Right, coronary heart disease. Now, risk factors for coronary heart disease: if you smoke, if you're obese, if you take certain medication, okay, if you have a poor diet, if you have coronary heart disease running in your family, you've got increased risk of getting coronary heart disease. Okay, remember, it's not one that passes on, it's one that kind of develops. And again, you need to be familiar with stents. So these are those like little metal crisscross tubey things that we put in heart surgery into the artery that might have become blocked. And then we're going to put a balloon inside and open up the stent, so we widen the artery. It doesn't actually remove the blockage, and then you've got all the risks of an operation, infection, general anesthetic, etc. You could replace the heart. So we need to find a heart donor, someone who's recently passed away of a similar age, probably, to try and have a donation of a heart. It's obviously a major operation. You will be asked to take immunosuppressants, which means it reduces the risk of your body rejecting the replacement heart, okay, because obviously that's not part of you, so your body might attack it, your immune system might attack it. And then your statins down the bottom is if you have particularly issues with cholesterol linked to coronary heart disease, statins can help to break down that buildup of cholesterols. There's obviously a side effect risk, and we've got to remember to take the tablet. So for higher tier, know what the treatments are, the risk factors for coronary heart disease, and know how to talk about the pros and the cons of each, because that is a skill that's going to be required there. Okay. Righty-ho, let's see. Okay, so what is coronary heart disease then? Again, a healthy coronary artery would look like this, where you have a nice clear lumen for the blood to pass through. And now your coronary artery, actually, I should say, it is, if you look at the heart on the outside, right? So you've not dissected it, you've got a whole heart, you've just pulled it out of an animal or a person. Around the outside of the heart, you kind of see like this artery that sits across the front. Okay, that is the particular artery that supplies the heart, all those cells that make up the heart tissue. That coronary artery gives all the different parts of the heart oxygen. I hope I'm making sense. Okay, if you look at a diagram of the heart, you'll see what I mean. So with coronary heart disease, oh, I'm keeping track on the other slides here for us as well. Perfect. Okay, that's fine. So the coronary artery supplies the heart with oxygen. Okay, so what happens over time with poor diet, with smoking, alcohol, all the other risk factors we spoke about, is that these fatty deposits can build up in the coronary artery. And as these fatty deposits or the plaques build up, it can reduce blood flow in the artery. Now, if that happens, then that means the heart cells, or the muscle tissue that makes up the heart, won't get enough oxygen. If there's not enough oxygen getting to those cells, it can't do respiration, so there's not enough energy. And this is what leads to a heart attack. Often, if you have a complete blockage here as well, it will lead to a heart attack. Okay, so that is coronary heart disease. And okay, again, we've mentioned all of this, so I'm going to run through. Cancer is an uncontrolled growth of cells. It's when cell division, mitosis, does not stop. It involves the mitosis of normal body cells. Two types of tumors: benign tumors, they have a regular shape. It says they're not cancerous, and they're usually not removed unless completely necessary. Benign just means it's not actively dividing, it's just sort of sitting there. Okay, it's not growing, there's no metabolic activity going on. And often if you're looking at tumors in the brain, if you've got a benign tumor in the brain, think about the complications of surgery in getting into someone's brain. You've obviously got to get through the skull and do all that kind of stuff, and very carefully get to the tumor to remove it. It's sometimes not worth it if the tumor is benign. Okay, and as long as people are happy to monitor regularly, have regular scans for different tumors, and they're not doing anything, we're all good. However, the other type of tumor is called a malignant tumor, and this reminds me of that Maleficent movie. Okay, Malignant and Maleficent. So the malignant tumor has more of an irregular shape. This is actually cancerous, which means you've got actively dividing cells making up this tumor. Okay, and we need to treat it, otherwise eventually it will be fatal. Third bit of language here is metastasis. This is when cancer spreads. So this is what I was talking about earlier when we said you've got a clump of cells, so you've got your tumor, and a little clump of cells will break off, and they use the bloodstream to travel around the body, and then they can settle elsewhere in the body and invade other organs as well. Okay, so metastatic or metastasis is when this cancer is actually doing the spreading. You'll be talking about a malignant tumor when we're talking about metastasis happening. Okay. So, okay, the immune system here now is at the bottom of the slide. Fabulous. Okay. Gosh, the immune system in a nutshell. Defending against pathogens. So pathogens, if you remember, are the microorganisms that cause disease. We are normally talking about bacteria, fungi, and viruses. They're the three biggies. So bacteria and fungi can enter the body and produce toxins or poisons. Viruses can enter, viruses actually get into your cells and mutate the DNA. And this is why when you get sick with a virus, it does make you really, really sick. So think about COVID, think about the flu, think about a cold, sometimes can be really heavy colds. They are all viral diseases. Chickenpox is another good one, right? They're all viral, and you won't just feel rubbish, you'll feel really bad, like muscles hurt, you've got a fever. That's because viruses have got into your cells and they're hijacking your cells. They're mutating the DNA of the cells to replicate themselves to make more virus. Bacteria and fungi, however, just tend to float around in the body. So when you get a bacterial chest infection, okay, what you're getting is loads of bacteria that are dividing, and your doctor will give you antibiotics for bacterial infections specifically. You never ever use antibiotics for viral infections, just bacterial infections. Now, how can we stop these pathogens getting in? Okay, and these are the ones you need to know about: how to stop pathogens getting into your body. Your skin is the biggest one. It acts as a kind of physical barrier. The trachea, so your throat here, the cells in the trachea produce mucus, they're called goblet cells, and they're lined with these hairs called cilia to try and trap any pathogens before they enter the rest of the body. Your stomach contains acid, so any pathogens that do get past the trachea end up in your stomach. Hopefully the acid will be able to get rid of those pathogens. In your blood, you have these cells that form platelets at wound sites to stop any pathogens getting in, to kind of form a covering or a seal. And in your nose, you've got hairs that also trap pathogens. Okay. Down the bottom here, your white blood cells in your immune system here, they produce antibodies and antitoxins to neutralize the pathogen. Remember that an antibody is just that it's a Y-shaped molecule, okay? And it's just a label that your body puts onto an invading cell, a pathogen, so that other white blood cells can come along to engulf the pathogen. And the antitoxins are like the poisons that the pathogen are producing, the antitoxins do the neutralizing of those poisons. So they don't make you feel ill, hopefully, reducing symptoms. Phagocytosis is the process where your white blood cells are coming along and they're engulfing the pathogen, taking it in, and then digesting it down completely to get rid of it. Right, these are the common diseases that you're going to need to know about. So in terms of viral diseases, we'll do viral first, and then we'll look at we'll do viral, then probably bacteria, and then we'll do fungal at the end. Viral diseases in humans, yes, humans, or in animals, I should say. You've got measles first of all, caused by a virus. Symptoms: fever, your skin goes red, you've got that blotchiness. It says treatment with vaccination. Treatment would be you would treat the symptoms of it, but actually preventative treatment would be the vaccinations. HIV is down here. This is again a virus in animals. It is a flu-like illness. There is no treatment for HIV because once you get HIV, it eventually, usually after many years, goes on and changes into another illness called AIDS. But HIV is a virus that attacks your immune system. Okay, and so what happens is, because it's been attacking your immune system, you get to a point where you get something like a cold, you get something like the flu, and then that completely can kill you because your immune system can't fight back. We use these drugs called antiretroviral drugs to control HIV. So just be familiar with that language. Your plant virus example is down here, which is TMV, tobacco mosaic. Look at the words there. The mosaic bit means that in plant leaves, you end up with a mosaic pattern that is actually discolored, which means it's supposed to be green and it's not green. Usually it's like a whitey, yellowy color. Because you've lost that greenness in the plants, what happens is that photosynthesis can't happen, okay, and therefore the plant eventually dies. To treat tobacco mosaic, you need to remove the infected leaves, and usually you would say burn the leaves because there's virus still on there, and make sure that all the tools that you use have been disinfected to remove any traces of the virus. Right, I think I said was going to do bacteria next. Gonorrhea is a bacterial illness that usually is sexually transmitted in humans. You end up with a thick yellow-green discharge and pain when you pee. You need to use antibiotics and use barrier methods, so use like physical things like condoms when you're having sex to stop this bacterial infection being transmitted. Okay. The other bacterial infection is Salmonella, and this again, back in the day, used to be not a lot more common. It was all in the news, where you had eggs and chicken that were transmitting this Salmonella bacterium from chickens to humans. If you get poisoning by Salmonella bacterium, your symptoms: fever, abdominal cramps, vomiting, diarrhea. Just not very nice symptoms at all. A lot to do with your digestive system. But here they've said the symptoms will disappear on their own within 2 to 5 days. I think only in extreme circumstances would you look at giving antibiotics against the Salmonella. It usually is that if you've not cooked your chicken properly, for example, this may be a possibility. Bacteria. Okay, protists at the top. This is another good one. Malaria, again, quite a common illness passed on by protists, okay, which is just these tiny kind of quite small organisms that live in blood cells. The symptoms of malaria are high fever, and now the treatment here is to control the spread of the mosquitoes. So try and stop them breeding. So mosquitoes like to breed where there's pockets of water, still water, like drain pipes and puddles and stuff like that, especially when it's warm. If we can get rid of those still bodies of water and also use nets when you're sleeping, that is a way to control the spread of malaria. Because malaria, the parasite will come along, suck the blood of a person, the mosquito will come along, let's get it right. They'll suck the blood of a person that has malaria, and in that blood is the protist. They will then fly out the window to another house and then suck the blood of another person because they want to feed off the blood. And the protist will get passed on from person 1 to person 2. And that is how malaria spreads so quickly. And lastly, there's only one fungal one, fungi. Rose black spot, again, use the name to help you remember. This is where you have purple or black spots on the leaves of usually rose plants. It means the leaves will eventually turn yellow and fall off. Okay, I'll just answer this message. Oh, press the wrong thing. And also stunted growth of the plant, again, because the leaves are turning yellow, there's no photosynthesis. If there's no photosynthesis, then the plant can't grow. Here, because it's a fungal infection, we want to use fungicides and remove all the affected leaves. Again, burn the affected leaves so we don't have any fungus passing on. Okay. Take a breath. So they put down here, communicable diseases in terms of a definition. Again, look at the word there to help you. They can spread from one individual to another. They are caused by pathogens. Okay. And that's what all of these are. So make sure that you're familiar with each of these. We're going to go up, up, up. We're getting there. Right. Where are we at with vaccinations? Okay, awesome. So we have, this is, I think, topic 3 now. If an individual is having a vaccination, this leads to what we call immunization or immunity against a disease. So what's the process? We inject an individual with a weak or inactive pathogen. That definition is important. Your white blood cells now think that your body's being attacked by that pathogen, and they start to produce antibodies, remembering that an antibody is just a label that goes onto that pathogen cell so that other white blood cells can attack it. The memory cells in your body will remember the shape of the antibody once it has been produced. So when the live form of the pathogen comes along, so when you're exposed to it, so say you've had your COVID vaccination and you're exposed to COVID, your white blood cells instantly recognize it and they produce the antibodies to label that virus very, very quickly and in a lot higher numbers really quickly. So it's like, bam, the whole armies come in as soon as you have any inkling of the actual real disease. And this is how we get our immunity or immunization against the pathogen. Okay, so it's quite this is why vaccinations are really useful. Now, in a situation where you have a lot of people in a big area, so think about big city or even the UK, this herd vaccination is where you have a vaccination program introduced across the country. Okay, and what the government do is say everyone has to get vaccinated. And so in this situation, if this purple dots, each of the purple dots was a person, okay, most people have had the vaccination and so they're immune. And so if this guy here with the black dot has is unvaccinated for whatever reason, because you have the herd immunity, it actually reduces the chances of him being exposed to the pathogen. Okay, so herd, think of like all the people as like a herd of people that are all vaccinated. Okay, sometimes we call it herd immunity, and this is what Boris Johnson was banging on about a little while ago over COVID, because he just said, "Oh, let them all become naturally immune by herd immunity," i.e., we don't want to roll out a vaccination program. Ridiculous, man. Okay, right, respiration. Okay, so now we're heading to the tail end. You'd be glad to know. Okay, it's a big old slog, but that's okay. Are we okay, Emily?
Yeah, I'm fine.
Yeah, okay. We've just got another. Okay, thanks, Emma. See you later. I'll send you the recording through. Don't worry. Okay, I'm just gonna take a little swig of drink here.
Okay.
Right then, respiration then. Now, this is a bioenergetics topic 4. It happens in the mitochondria. Okay, so that's the organelle that we're looking at. The formula here you will need to know, including how to balance it. So oxygen, we take it in by breathing in. Glucose, we take it in by eating. Okay, that forms carbon dioxide. We breathe it out, plus water. Well, we pee. Hopefully we pee. And then that also produces we should say it transfers energy. Don't say it produces energy, it transfers energy. Energy is not strictly a product, but it is actually the point of respiration. Now, that energy enables us to maintain our body temperature. It enables us to move, so muscle contraction. So that's really important to mention as well. Okay, 2 types of respiration you need to know about. You've got aerobic and anaerobic. Okay, so aerobic is the respiration that it says produces energy, takes place in mitochondria. It's more efficient. Aerobic requires oxygen to do respiration. It's that equation we just looked at that's happening again and again. Plenty of oxygen around with aerobic. It produces carbon dioxide and water, again, like we've just said in the previous slide. Now, anaerobic respiration also produces energy, but anaerobic is what happens when there is less oxygen around. Okay. Now, the amount of energy that's produced compared to aerobic is much, much smaller, but it still does produce energy regardless. Okay. And that's why they've said it's less efficient. The product of anaerobic respiration is lactic acid, and this is what will cause cramps in your muscles. Okay. Now, in yeast, anaerobic respiration goes like this. Your glucose is converted into carbon dioxide plus ethanol plus energy. And what we can do is we can use both of these products to do useful stuff. Your carbon dioxide can be used to make bread rise, which is why when you bake bread, you add a little sachet of yeast. And the ethanol is alcohol like that we can drink, and this is used in brewing or in wines and in spirits. Okay, and this is why yeast is important. We can also sometimes we talk about fermentation as well. Okay, let's go to here. Right, lactic acid. So this is that thing that's produced when we have anaerobic respiration happening. And so lactic acid, when it builds up, it can cause both cramp and muscle fatigue. At higher tier, we're talking about, what's it say here? It says blood flows through the muscles and transports lactic acid to the liver where it's broken down. So you're running a race, you get to the end of the race, you've got this buildup of lactic acid, your muscles are sore, you might have a cramp. That lactic acid goes to the liver, and in the liver, it gets fully broken down into carbon dioxide and water. Okay, it just requires some time to do that. Now, the oxygen debt, again, higher tier, is the amount of oxygen the body needs after exercise to break down and remove all the lactic acid. Okay, so while you were making the lactic acid, you didn't have enough oxygen getting to the cells, and then once you stopped exercise, you started breathing hard and catching your breath. Okay, that oxygen debt, you're going to bring oxygen along to fully break down the lactic acid. If it builds up, it's going to be painful. Okay. Right, your metabolism. So the definition of metabolism is all the chemical reactions that happen in your body. In your body, what's happening is you're converting glucose into storage molecules like starch in plants, glycogen in animals, and cellulose that's used for plant cell walls. We can use some of these metabolic reactions to form lipids where we're joining together 1 glycerol and 3 fatty acids. We can use the glucose from respiration and nitrates, put them together and form amino acids. Quite useful because amino acids make proteins. We can use some of the glucose in the process of respiration, and we can also break down excess protein. So we can chop up any excess protein in your body to form this stuff called urea. And this is why when you eat loads of meat and then you wake up the next morning, if you had loads of meat for dinner, you'll find your urine that your urea is made from is quite strong because you've got a lot of protein in it where you've eaten a lot of meat. That's your metabolism. Okay, home run now, and then we'll have a quick look at required practicals. So, photosynthesis. This is what all plants do. It happens in the chlorophyll, which is stored in the chloroplasts. Photosynthesis is an endothermic reaction, which means that energy is taken in from the environment for the reaction to happen, and that's the energy is taken in through light. Okay, and the light energy is actually converted to what we call converted to glucose, which is what we can describe as a chemical energy store. So Plants take in your carbon dioxide from the stomata of the leaf. They take in water from the roots of the plant. They form oxygen, which is removed through the stomata as a byproduct, and they form glucose, which is used for things that we'll talk about in a sec. And then also you've got, it says energy here as well. I don't think strictly you need to have the energy on here. Okay, you probably haven't learned it as this energy. You need to write the word light onto the top of this equation, okay, or sometimes chlorophyll onto the arrow rather. So the glucose that's made in photosynthesis, well, we can use this glucose for respiration at nighttime when there's no light. We can store some of the glucose as starch. So if the glucose is a paperclip, we can hook it together to make starch. We can use some of the glucose to build amino acids for proteins, as we said on the previous metabolism slide. We can build fatty acids for lipids. And we can also use glucose to make cellulose for growth of the cell and to strengthen the cell wall. Yes, my darling. Come in. Sorry, we've got a visitor. Come in, darling. What's the matter? Sorry, excuse me. It's always going to happen, isn't it? Yes, darling. Well, I am teaching a lesson, okay? All the children need to do their learning. Right, we've got a visitor. Oh, mind my wire, please. Okay, mind your head. Oh, here we go. Right, sit on this leg here for me. Okay, so with photosynthesis, now we've got the basics in place. I've got to adjust this headset. You've got your limiting factors of photosynthesis, and these are the 3 graphs that you're going to need to know. So you're expected to know what happens when, to the rate of photosynthesis, when we change temperature, when we change the light intensity, and when we change the carbon dioxide concentration. So on every graph that you have here, up the side is the rate of photosynthesis, and across the bottom, we've got our 3 different variables here. So with temperature, this is the one that's different from the other 2. And this, if this comes up and you're describing this graph, you start from the bottom here. You can see, obviously, as we increase the temperature, we're increasing the rate of photosynthesis until we reach the optimum temperature. And beyond the optimum temperature, the enzymes become denatured, and this is where the reaction goes down to 0. Okay, you must talk about enzymes when you're talking about this temperature graph. Okay, really important. And that's also where you can include, as the enzymes become denatured, the active site changes shape, and therefore is no longer complementary to the product. Stop. Right. Next 2 graphs both have exactly the same shape. Okay, and so let's look at light intensity first. So the first graph here, as we increase the light intensity, the rate of photosynthesis is increasing. Okay, and you get to a point where the let's remove that. Here you go. You can draw on that. You get to a point where we have a flat line here where the rate begins to stay the same. Now, the question will ask you to look at, well, what's happening here on this flat line bit? Well, we know that as we come across this in this direction and along this axis, we're actually increasing the light intensity more and more and more and more. Okay, and so there's not, so light is not limiting our rate of photosynthesis. So it must be something else. Okay, and so we could say that you've got carbon dioxide that is limiting the rate. You could also say the temperature might not be quite right. Okay, and could be limiting the rate. You could also say there might not be enough water for photosynthesis to happen, but definitely light is not limiting the rate here in the middle. Now, come down, can pick it up here. The last graph here is the carbon dioxide concentration, same pattern. As we increase the carbon dioxide concentration, the rate of photosynthesis increases, and then the rate stays the same, levels off, plateaus, stays constant. Now, this is because it's not, we don't have a limiting factor of carbon dioxide because we've got plenty of carbon dioxide here, but we may have light intensity limiting the rate, or we may have a problem with temperature, or we could also say that water could potentially be limiting the rate of photosynthesis. Okay, so that's how you would kind of describe these graphs. When you're talking about explaining the graphs, you've got to tell me why it looks like that. Use the word because in your answer. Okay, sit down. I'm teaching, okay, so I need you to play. Right, if you have a greenhouse in your back garden, or if you've ever been in a greenhouse, the point of them is to maximize photosynthesis. So what we could do, what we want to do is to maximize photosynthesis, we need to get the conditions exactly right. So they could use oil burners to keep the greenhouse warm, to keep our, come off here. Can you still hear me, Emily?
Yeah.
Perfect, okay. Oh, hang on, I don't know what's now going on. Bear with me, something's just dropped out.
That's okay.
Here we go. Can you see the screen now?
Yeah.
Fab. We're getting there. Are you okay? It's a bit of a hard slog, but you're all right.
Yeah. It's all good.
Fab, good. Okay, we're nearly there, nearly there. Let's go. So if we use oil burners here, it means we can keep our temperature correct, and also burning oil would release carbon dioxide, which is one of our substrates that we need for photosynthesis. Right, yeah. Wait, I'll come to you in a minute. I'm teaching now, honey, okay? No, I actually don't have time for it. Okay? Right, obviously greenhouses being clear, they allow more light to be absorbed, and we can pump in more water so that we have plenty of water and carbon dioxide to maximize the rate of photosynthesis. So what's the point of this? What we're doing with our greenhouse is we want to maximize the yield, so whatever we're growing, to maximize how much we grow, and the profit that we make, so the money that we're making from, assuming it's fruit and vegetables that you're growing. But they are also very expensive to run and maintain. So talk about positives and negatives of the greenhouses. Okay. Right, where are we at? Perfect. Right, honey, stop your noise, please. Okay. Drive me bananas. So you need to know about how stuff moves around plants. So with xylem vessels, these are the vascular tissue, the tubes, if you like, and so are phloem actually are tubes. But in the xylem, you have water and minerals carried. Roche, could you stop, please? You have no end walls between the cells. The cells these are actually dead cells that the xylem's made of. We said water and minerals carried. They are thick walls that are stiffened with this stuff called lignin to make them really strong. Think of them as a really strong straw. Okay. You then have your phloem vessels here. So this is where your food is carried, also dissolved in water. Okay. So phloem and food, think about that. Right. Are you now going to leave this room? Because all I can hear is your clicking and it's really annoying me. Okay, if you can sit quietly, you can stay. Otherwise, I'll be asking you to leave. All right, lady? So with the phloem, you have walls that have perforations on the end. These are called sieve plates, okay? And also important to remember is in phloem, water and food moves in two directions, okay? Now, language. When you're talking about the movement of water, you're usually talking about transpiration. Whereas if you look on the board, translocation is how dissolved sugars move through the plant. Okay, so translocation is the phloem you're talking about, and transpiration is usually the xylem you're talking about with water. Okay, right. This structure of the leaf, this will probably take you back to like maybe year 7, 8, maybe even year 9. My goodness, there we go. It's done. Go and play, please. So your structure of the leaf here, you've got this, it's like a cake. Your top layer here with your waxy cuticle that reduces water loss, And right down the bottom, again, you've got a waxy cuticle, again, to reduce water loss. You've got your stoma at the bottom that are bound by guard cells, and that allows gases in and out. It allows carbon dioxide to come in and oxygen that's been formed in photosynthesis to be removed. Also, you've got water in these air spaces here, which is really important. At the top here, you've got this upper epidermis, and you've got a layer of cells that don't really do anything. It's just a layer of cells at the top. You then have these palisade cells that are stacked up next to each other, and these are like soldiers all stood in a row. These palisade mesophyll cells have chloroplasts in them that can actually move, and the chloroplast, because that's where photosynthesis happens, the chloroplasts will orientate themselves so they are in a position to maximize absorption of light for photosynthesis. In this middle section here, you've got this mesophyll, spongy mesophyll layer that is made from these round mesophyll cells. And in between them, you have plenty of air spaces around. And again, in the air spaces, you will find carbon dioxide, you will find oxygen, and you will find water as well. Okay, so this is your basic structure of the cross section of a leaf. And then if we come down, we've got, right, this idea of transpiration stream. And this, think of this as the water that starts off in the roots, and like a column of water, like a piece of string, starting off at the roots, going all the way up the roots, up the stem, across the leaf, and then out through the stomata of the leaf. Okay, you've got a long single column of water. And so the definition of transpiration stream is the loss of water through the stomata in the leaves. Here, we've got the guard cells open, the stomata, so those two guard cells that were bounding the stomata. Yes, you can go. And the water vapor escapes. So with transpiration stream, we're talking about water specifically. As the water escapes, it's like pulling up this piece of string, and the root hair cells in the roots will absorb more water. Now this, I'm going to shut this door here because now we've got peace and quiet again. Excuse me. This number 3 here now creates what we call a concentration gradient. So there's more water in the roots now than the leaves. And so by osmosis now, the water is going up the xylem, which is in the stem, all the way to the leaves. And you have this constant repetition of this process. Again, I think of the water stream like a long piece of string. And as we've got transpiration from the stomata, it's like pulling the piece of string, and that means that more water can come in from the roots, okay, constantly. So key ideas with transpiration. When we increase the temperature, we increase transpiration rate. When we increase the light intensity, we increase the rate of transpiration. When we increase the wind, so on a really windy day, more water is carried away off the surface of the leaf, so we increase the transpiration rate. And if we increase humidity, so if it's a really muggy, humid day, that means there's more water in the air, okay, so more water around the leaves. This will actually decrease the rate of transpiration because we no longer have a concentration gradient that's as big, because you've got water outside the leaf and water inside the leaf. So how far are we? Okay, so we've got stem cells. I'll tell you how many we've got left, just so we can pace ourselves correctly. Dub, 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 dub. Perfect. Lovely. So we've got, right, you'll be glad to know, we've got 2 more slides to do, and then we're done with content. Okay. So is that all right?
Yeah, that's fine.
Perfect. So we've got undifferentiated cells that can specialize to become any type of cell. That is the definition of a stem cell. Okay. What you're thinking about is a cell that can become anything that it wants to be, right? It's like its destiny is not yet decided. Where we find stem cells is in embryos, so embryonic stem cells, and then also in the bone marrow. These are your adult stem cells. okay, in humans. There's definitely ethical issues here, and you might be asked to evaluate the embryonic or the adult stem cell treatments and give some kind of a conclusion. Remembering in an evaluate question, give me the pros, the positives, give me the cons, the negatives, and overall, tell me what you think. Okay, that's how you structure an evaluate question. So we could use stem cells to cure diabetes, okay, which means that we would be adding stem cells to the pancreas to produce insulin-producing cells. And we can also cure paralysis, again, by introducing stem cells with nerve cells around them, so in the spinal cord, for example, and so more nerve cells would be generated. In plants, we find stem cells in the meristem tissue. This kind of question comes up not very often, but it is often one that's lost on people sometimes. Meristematic or meristem tissue in plants contains stem cells, and they are in the roots, in the fruits, and in the shoots, and also in the very, very tips, okay, of plants. So any bit that really is growing and developing of a plant will have meristem tissue that contains stem cells. We can use implant stem cells to produce clones very quickly. And so this means that it should say rare species, protect rare species from extinction. And we can also produce large numbers of plants that are resistant to certain diseases. Okay, so if you've got a crop of carrots, for example, we can produce carrots that are resistant to common whatever diseases that carrots might get. And this, I'm glad to say, slide number 54, we made it, is therapeutic cloning, the last idea here, okay? And so here is where you have a human egg cell that is taken from a donor, okay? So I take an egg cell from a person, we're gonna remove the nucleus and we're gonna get rid of it, okay? We stimulate cell division, okay? And that means that that cell will start to develop into an embryo. So this kind of mitosis is happening. Then the stem cells are removed after a couple of days. And then what we do is we use those stem cells from the embryo for therapeutic use. So you might be putting it into a patient, to someone who's had a car crash, for example, and we give them stem cells in their nervous system to produce more nerve cells, okay, to cure paralysis potentially. At the very top, what I didn't say was, okay, you've got a nucleus that's removed from a patient cell, and we're going to put that nucleus into a donor cell. Again, I don't really like this diagram very much, but that nucleus goes into a donor cell and to combine those two together and stimulate cell division. This is discussed. There's a paper 2 section where this goes into a lot more detail about cloning here. But for this particular section, make sure for higher tier that you know about ethical, social, and clinical issues surrounding the use of stem cells in therapeutic cloning. Embryonic stem cells pretty much are illegal at the moment because of the moral, the ethical, the social issues surrounding them. Okay, so be able to give me some positives and negatives of using stem cells in therapeutic cloning. Okay, right, Amelie, how are you doing?
Yeah, I'm doing good.
Okay, right. Are you happy to go on to having a look at some of the required practicals?
Yeah, that's fine.
Yeah. Okay. Again, if you need to go, let me know. What I will do is carry on recording, and then I can just pop over the recording to you. You can come back to it. Let's have a
I'll
look.
probably need to leave at around 12.
That's absolutely fine. I wasn't expecting it to take this long. Where are we at? 47. Let's try and do this in 15 minutes, shall we? Let's get it done. Okay. So first idea here, first required practical, beginning of year 9. It is, let's make this a bit bigger so we can see it a bit more nicely. Oh, a bit much. This is your microscopy. Okay, you need to be able to label the parts of the microscope that you see on the board. So just go through and remind yourself of what's what. Okay, remembering the stage is the bit the slide goes on. And then you've got your coarse focus and your fine focus knobs there. Coarse focus is for big movements, so the stage up and down. Fine focus is for smaller movements. Converting your units, we mentioned earlier on, if we're converting from millimeters to micrometers, you're timesing by 1,000, don't forget that. And your triangle here, IAM, image size, so the picture that you see, that you're given in the exam. The actual size, so the real size of the thing that you're looking at, probably in micrometers, and then your M is your magnification. Okay, magnification doesn't have a unit, it is just times 10, times 40, times whatever. When you're studying an animal cell for this practical, you're going to use a dye called methylene blue. That was what I don't know if you guys did I think you were in COVID. We scrape the inside of your cheek cells to find animal cells and make slides out of them. We would use methylene blue. If you're looking at plant cells, this would be the onion skin that you did, and that is using a dye called iodine. Okay, comes up orange. Next, food test, okay, or food testing. There are different food tests that you need to know, and there's one additional one that I'll tell you about for the fats down here. If you're testing for iodine, you're going to add starch to the food, potatoes, crisp, rice. It changes from orange to blue-black. It's a nice image here to remind you. If you're testing for glucose, the presence of glucose in a food, you're using Benedict's reagent. Benedict's is the one that we have to heat when we use it. It changes from blue to orange, and sometimes it goes green. This is the only test that tells you how much sugar is in the solution, how much glucose is in the solution. It is the only quantitative test that you will do. Okay, tells you the quantity. The red is the most sugar in the solution, high concentration of glucose. The Biuret solution is what you use to test for protein, and that is again blue to start off with, and in the presence of protein turns pink, or actually I remember purple, this beautiful purple color. Okay, so Biuret's protein purple. Sudan 3, and you may not have used this because I don't use it often in the lab, test for lipids. This is where you have a red layer forming on top when you add Sudan 3 to a solution of lipids. What you probably will have done for lipids is the emulsion test using ethanol and water and then shaken up your solution. That goes from clear to a cloudy emulsion. That's your food testing. Told you it's going to be whistle stop. Right, the effect of pH on amylase. So here you have taken different pH solutions, okay? And what you're doing is you're adding this enzyme amylase to what are you adding it to? Let's have a look. Come here, yep. You're adding it to a starch solution. I'll show you the whole thing here, probably easier like that. So the independent variable, the thing you are changing is the pH of the amylase. The dependent variable, what you're recording in your spotting tile here, is the time taken to break down the starch. So when you start off at the beginning, you should see starch is there. You should see blue, black, blue, black, blue, black. As the starch is broken down into glucose, eventually the color will change to an orangey color where you've got no starch left in the solution because amylase has fully broken down the starch into glucose. The control variables, the things we want to keep the same, is the volume of pH and the volume of starch. Don't say the word amount, say volume. The temperature that you are using for the experiment, we want to keep the same, and we're going to use a water bath to do that. Okay. Explanations. The reaction will not work at an extreme pH as the enzyme will have become denatured. Again, same idea to do with enzymes that we spoke about previously. The amylase breaks down starch to produce glucose, and then they could link food tests to this as well. So be aware, obviously, the iodine that we've spoken about. So in the practical, we are only measuring every 30 seconds. So what you do is you get the enzyme and you get the starch and you let the enzyme break down the starch. Every 30 seconds, you take out a drop and put it in your spotting tile with iodine. Now, we're only measuring every 30 seconds. What we need to do to improve it is to measure at more regular intervals, maybe every 15 seconds. We should use smaller intervals of pH to give more accurate results. You could use pH 4.1, 4.2, 4.3 if you wish. Right, we've now got osmosis. I'll tell you what we've got left. We've got osmosis left. We've got pondweed. I'm sure pondweed is the last. We're nearly there, Emily. We're nearly there. Go back to here. So osmosis, this is the, remember osmosis is how water moves from a high water concentration to a low water concentration. We had our test tubes or our beakers, and we have these cylinders of potato that we put into the different solutions. And we see here we've got no solute in this one, and you've got 3 dots here. You've got a few more, a few more, and a few more. So we've got, no, we haven't. 1, 2, 3. 1, 2, 3. 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5, 6. I'm not quite sure what's happening here. But you're putting your cylinder of potato into different concentrations of solution. And again, we'll come down here for a more detailed look at what's going on in this practical. So we are changing, independent variable, the concentration of sugar solution. We are measuring the change in mass, okay? Change in mass of the potato. We are controlling or keeping the same the volume of solution, not amount, the length of the potato chip, making sure we cut off the ends, the time that we left it for, so we might have left them in the solution for 20 minutes, and the temperature of the experiment we want to keep all the same and constant. Excuse me. This was what I mentioned earlier a few years ago. They asked students questions not to do with potatoes, but to do with carrots. And for some reason, students got really confused. The principles were exactly the same. Water moves in through osmosis if you put the potato in a low concentration sugar solution, because there's more water outside, so the water's going into the potato. Water moves out through osmosis if the potato is placed in a high concentration sugar solution. So if you put it in a very high concentration solution, there's more water in the potato than outside, so the water starts to move out and your mass will go down. We need to be able to work out the percentage change in mass, okay? So you do your, let's get this right, final minus initial mass divided by the initial, and then we can times it by 100. Remember to dry the chip before you weigh it to make sure there's no excess water that would affect the mass that we measure on the balance. Lastly, required practical for bioenergetics is your photosynthesis one with pondweed. This is a lovely, lovely experiment. So you're going to put your pondweed in your test tube. You're going to add water. You're going to add sodium hydrogen carbonate to put carbon dioxide into the water. And you're going to have a lamp that you're going to move away at set distances. And we're going to count the number of oxygen bubbles produced by the pondweed. So how would you set up the experiment? Well, we're changing the distance of the lamp from the pondweed, i.e. the light intensity. We are counting, dependent variable, the number of bubbles, usually produced per minute will be the thing that you count. Control variables, the volume of sodium carbonate, which is the stuff you put into the water to put carbon dioxide into it. The concentration of sodium carbonate that you use, you're using the same. Excuse me. And the temperature that the practical is at. We want to keep the temperature constant. We don't want that to affect our results. We've said sodium carbonate acts as a source of carbon dioxide. Temperature, we've said, can speed up the reaction or can denature the enzymes. So the number of bubbles tells you about the rate of photosynthesis. The more bubbles there are, the greater the rate of photosynthesis. And lastly, the inverse square law you might be asked to use. Light intensity is proportional to 1 over the distance squared. So as you double the as you double the distance, the light intensity will fall by 4. You might be given this and asked to put numbers into it. That's the only thing to have a think about and have a practice with your workbook or with your revision guide that you're using. Right, we are just before 12. How do you feel now, Amelie? I hope that wasn't too quick.
No, that was good, yeah.
Good, brilliant. So I'm going to just stop this share. So that was everything for paper 1. If there is anything you need between now and Friday, if you give me a shout or give Nirjan a shout and let him know, and we'll get you sorted. Do you have any questions at this point?
I was just wondering if maybe you
Oh, I've lost you.
could send the, you know, the lungs